Evening, everyone. Welcome to the webinar series. It's a warm Saturday afternoon here in India, and I hope it's a pleasant weather in Singapore out there. And good morning to all the Western world people joining us. Uh, we are starting a webinar series, namely Stem Cells from Basics to Clinical Application. In that, we have an excellent line of speakers who will be sharing some deeper insights on their recent research related to various domains of stem cell biology, current state of knowledge, and its impact on future application. With this webinar series, we intend to help the budding researchers gain better understanding on stem cell research. For our today's session, we have Dr. Srikala from SRIS. I would request Dr. Shravanti Rampalli to please welcome our today's speaker, Dr. Srikala Raghavan. Over to you, Dr. Shravanti.
Thank you, Pooja. As Pooja said, this is our first initiative at IGIB to have a stem cell specific seminar series. Welcome you all. Today's speaker is a very good friend of mine and an excellent scientist, Dr. Srikala Raghavan. I have known her for over a decade now, and I can tell a lot of scientific and non-scientific things about her, but let's stick to scientific stuff today. Srikala obtained her PhD from University of Cambridge. Then she did her postdoc with Dr. Elaine Fuchs. She was at Chicago. Later, Elaine moved to Rockefeller, and Srikala moved along with Elaine to Rockefeller to complete her postdoctoral studies. Srikala then joined Columbia University, New York, at Dental College Medicine. She started her own independent lab there. She spent almost 5 to 7 years over there, and then she was recruited to INSTEM Bangalore to initiate a program on Center for Inflammation and Tissue Homeostasis. Srikala joined INSTEM as an associate professor. She spent a decade over there, and now she has moved to Singapore. She is a principal investigator at CSIR-IGIB, and she continues her interest on stem cells in the area of skin or organ, and she's doing some really fantastic work, offline projects also, and what I know of her work, what she's going to present today. And I welcome Srikala here, and thank you for accepting our invitation. Please go ahead and talk about your research.
Dr. Raghavan, before you start, just a few housekeeping things I would request to our audience. So we will have a Q&A session post-talk. You can please post your question in the chat box. Over to you, Dr. Raghavan.
All right, thank you so much, Pooja, and thank you, Shravanti, for this for your invitation. I'm very excited to talk to all of you. So let me just share my screen. All right. Just trying to. Okay. All right. So good evening to everyone from Singapore. It's I guess it's closer to night here. And again, thank you very much for this opportunity to tell you a little bit about the work that's happening in our laboratory. So today I'm going to tell you a story which is about how mechanical signals are critical in maintaining quiescence of the hair follicle stem cells or the multipotent stem cells in the skin, known as the bulge stem cells. So in my lab, we use the mammalian skin as a model system to address questions related to immune regulation and epithelial homeostasis in various surfaces. So the skin is a fantastic organ to study this because, of course, it's the most external organ. It's also our largest organ. And it has a wide repertoire of stem cells, and I will talk about that just in the next slide. But before that, just to give you a little bit of introduction. So the skin consists of an outermost epidermis and then a deeper dermis. In development, the epidermis arises from the embryonic ectoderm, and the dermis arises from the embryonic mesoderm. And the epidermis and dermis are kept together by this acellular matrix known as the basement membrane, which is comprised of extracellular matrix molecules, which are secreted both by the epidermal cells, which are known as keratinocytes, as well as the dermal cells known as fibroblasts. In addition to the epidermis and the dermis, the skin also contains a variety of appendages, the major one being the hair follicle, which is represented here. And the hair follicle is associated with this gland called the sebaceous gland. And this is important for producing an oily substance known as sebum. And in addition, there's also, of course, sweat glands, which is very important for thermoregulation because, of course, the skin is critically important for one of the critical functions it performs is thermoregulation. What I'm not going to talk about today is that the skin also has a very wide repertoire of immune cells because, of course, being the most external organ, it is exposed to the environment and therefore is critical. One of the main role it plays is defense. And so I mentioned the stem cells within the skin. So they come in two flavors. So there are stem cells that are found within the epithelia itself. And so the basal layer of this epithelia are proliferating and everything else beyond that is post-mitotic. And so there are pools of stem cells which are marked by integrins, which are found within the basal layer of the epidermis. These are unipotent. So if these are recruited or these come into effect when you have to replenish the skin itself, the epidermal compartment itself. And then you have other sources of stem cells within the skin, and those are found within this region of the hair follicle known as the infundibulum, and also region of this hair follicle known as the bulge. So over the past 30 years or so, there's been a huge amount of interest in this particular compartment, the bulge compartment, partly because this has been shown to be the source of your multipotent stem cells. So cells from within the bulge compartment not only can give rise to hair follicles, which is the organ, the appendage in which they are found, but under conditions of wound healing, they can also give rise to the epithelia itself. And so there's been a tremendous amount of interest in understanding not just the properties of the stem cells, but also the niche factors that are important in maintaining the stem cell, these stem cell pools. And so one of the, in homeostasis, what these bulge stem cells do is to give rise to hair follicles. And so within this hair follicle, these stem cells will then get mobilized and they'll go through these phases. So the hair follicles go through distinct phases. It's known as the anagen, the catagen, and the telogen. And so the anagen is the growth phase where stem cells get activated and contribute to the rapid development of the hair follicle and the hair shaft. And then at some point, the lower 2/3 of the hair follicles get destroyed, and that's known as catagen. And eventually you have the telogen phase, which is when the stem cells are sitting in that quiescent phase. I see that there are questions. So do you want me to answer questions while I'm doing my talk or at the end?
At the end, Srikala, you can take questions at the end.
Okay, all right. All right. So as I mentioned before, a lot has been known over the past 30 years about cell intrinsic factors. So these are a bunch of transcription factors and other factors that are important in maintaining this quiescent pool of stem cells. However, much less is known about the niche factors. So, the cell-to-substratum adhesion or even cell-to-cell adhesion factors that may be responsible for maintaining this pool of stem cells. And so in today's talk, I'm going to tell you about the role of these cell-to-cell junctions in maintaining the stem cell quiescence. So shown over here is an electron micrograph from the bulge, exactly from this compartment. And in black are these junctions known as desmosomes. These are electron-dense structures, and these associate with an intermediate filament network known as the keratin network. And in between the desmosomes, you have the appearance of adherence junctions. And these are the junctions I'll be focused on today. So these are junctions that are nucleated through cadherin to cadherin, so homophilic cell adhesion. And then on the other end, they associate with the actin cytoskeleton network. And so we were very interested in trying to understand what really happens to these stem cell, the junctions of the epithelia, as this hair follicle is sort of cycling through these various phases. So as it's going through this growth phase or sort of the destruction and the rest phase. And so is there a change because in these stem cell, in the stem cell compartment vis-a-vis its intracellular adhesion? So to look at that, we decided to look at two different phases. One is the growth phase. So this is when the hair follicle is rapidly growing out. And so just to orient you, this is a section, or this is a single hair follicle. This is the bulge region. So it's marked in every single image. And these cells marked over here with the marker CD34, cell surface marker CD34, marks the bulge stem cell compartment. And these are the stem cells. And so as the cells are starting to proliferate, they start expressing the proliferation marker Ki67. So as I mentioned before, this pool of stem cells are actually quiescent. So they don't proliferate, but their progeny will proliferate. And so when you now start looking at the cell-to-cell junctions, so we looked at E-cadherin, alpha-catenin, and Benkillin. And what you can see is that there's a very strong expression of each of these proteins present within the bulk stem cell compartment. And very interestingly, these proteins sort of get, the expression of this gets diluted out as the cells are proliferating. Again, suggesting that cell junctions are pretty labile and they are sort of, the expression of some of these cell junction proteins are downregulated in rapidly proliferating cells. And that's not surprising because that's something that's already known from the literature. And so when you go back and look under an electron, using electron microscopy, you can see that in this sort of proliferative part of the hair follicles, there are fewer cell-to-cell junctions. And here we've actually just marked the desmosomes and used them as a proxy to mark the adherence junctions, because adherence junctions really are not visible in EM. And then when you go back and look at the stem cell during the quiescent phase, which is the telogen phase, as I mentioned, again, you can see that there's a very strong expression of CD34. There is no Ki67, so there's no proliferation at this stage. And then you can see a very beautiful expression of these cell adhesion molecules and much higher levels of expression. And this is associated with stronger cell-to-cell junctions. Again, suggesting that in fact, these junctions seem to be really critical in sort of maintaining this cell-to-cell boundaries in the quiescent phase. And so we were very interested in trying to understand what the role of these junctional proteins are, if anything, in the epithelial stem cell niche. The expression pattern suggests that they may be playing a role, so we wanted to go back and ask, is it really playing a role in this process? And so to address this, we decided to conditionally knock out two proteins that are part of this adherence junctions. The first protein we knocked out was Vinculin, and the second protein that we knocked out was Alphacatenin. And I will take you through the results that we got. So what is Vinculin? So Vinculin is a adapter protein. As you can see here, it associates with these proteins known as alpha-catenin and beta-catenin. It is found in these cell-to-cell adhesion junctions. It is also found in cell substratum junctions known as focal adhesions. The vinculin consists of a head and a tail domain, and it binds a variety of proteins, and then it has a hinge region. The really fascinating thing about Benkillin is that it's a mechanotransducer, meaning that it actually transmits forces. And in fact, Benkillin itself exists in two conformations. So it exists in this closed conformation where it's not in an active state. And then when it opens up, it's in an active state. And this unfolding of Benkillin actually requires force, approximately 2 piconewtons of force. And as I mentioned, Benkillin is found in two independent junctions. So it's found in these cell-to-cell junctions that I have marked over here, and is associated with the protein alpha-catenin, and associates on the other side with its to the actin cytoskeleton network. Benkillin is also present in cell-to-substratum junctions, which are nucleated by integrins. And again, in this situation, it functions, associates with the actin cytoskeleton network and functions as a force transducer. So Benkillin has been previously knocked out, and the global knockout of Benkillin results in embryonic lethality at about embryonic day 9.5. These animals die due to heart defects and failure of neural tube closure. So we got interested in trying to understand the role of Vinculin in skin. But before that, as I mentioned, I said that Vinculin is a mechanotransducer. So what do I mean by that? So Vinculin is present at both of these junctions. So to the cell substratum junctions nucleated by integrins and cell-to-cell junctions that are nucleated by cadherins. And at the level of the cell to substratum junction, Vinculin functions to basically amplify actomyosin contractility by associating with a protein called talin. And what it does then is it basically allows talin to sort of also unwind, very much like what Vinculin does itself. So talin unwinds, and by unwinding that, it basically exposes binding sites for F-actin. And so then this then recruits the cellular contractile machinery, and then this is what sort of generates forces at the cell to substratum junction. And likewise, at the level of the cell to cell junction, Vinculin is associated with this protein called alphacatenin. And then this also associates with the actin cytoskeleton network. The other thing that Vinculin does is it actually recruits a whole bunch of actin modulating enzymes like VASP and ARP23, and also helps in the polymerization of the actin cytoskeleton. And so once actin is polymerized, it actually generates forces. And these forces allow for cells to actually be able to come together and to form stable cell-to-cell junctions. So Vinculin functions at both junctions and is a critical mechanotransducer. So what is actually the function of Vinculin then? So we generated the tissue-specific or the conditional knockout of Vinculin in the skin. And you can do that using a skin-specific promoter known as the keratin 14 Cre. And these animals were actually quite surprisingly born looking completely normal in the correct Mendelian ratio. You can see this is at age postnatal day 53. And of course, we've generated a knockout because what you can see is that there is no expression of Vinculin either in the hair follicles or in the keratinocytes or by Western blots. So this told us that, in fact, these animals looked perfectly normal. So the only thing that we could tell right off the bat, and even if you're not a hair or skin aficionado, I think you would notice is that the hair in these animals looks slightly more sparse than its wild type glutamate control. And this phenotype is carried throughout the life of the animal. So even at 1 year old, you can see that these animals have sparser hair. So since these animals had sort of a hair defect, we decided to go ahead and look at the hair follicles, and the developing hair follicles. And this is exactly what I just explained to you. So it goes through sort of this anagen, catagen, and telogen. So you can see this beautifully, and this is the first hair cycle. So you can see that this is anagen, this is catagen, and this is telogen. So the hair follicle grows in size by 10-fold. Okay, so this is a 100 micron scale bar. So it goes from approximately 100, 150 microns all the way to a millimeter in size. And so there's a tremendous amount of growth that happens fueled by the stem cell compartment that I talked to you about. And so even if you're not a hair follicle expert, I think you would agree that the hair follicles in the Vinculin knockout did not look normal. So the first thing that we noticed was that, in fact, the hair follicles did not grow to the size of the wild type. And the second thing that we noticed was that, in fact, they were sort of going through all of their developmental stages faster. So, for example, if you have to go through anagen up to day 12, these guys were already going into catagen, which is the destructive phase, by day 10. And then they kind of catch up, they both catch up, and they both end up being in telogen, which is the resting phase, at exactly the same time. And so what we showed was that the hair follicles never reach the same size, and they're actually in different phases of the cell cycle. So we decided to, so this is work that was done by my graduate student, Ritushri, and then my project student, Sergio, and they basically took animals for 100 days and basically followed the hair follicle cycle. This was a tremendous amount of work. And this is just summarized here. And what we could show was that normally in the wild type, interestingly, the first two hair cycles, so this anagen, catagen, telogen, is completely synchronized. So no matter where you're looking at the back skin of the animals, you see exactly the same phenotype with respect to the hair follicles. And so there is so initially the first telogen starts at about postnatal day 21, and then so it's very small, and then it goes back immediately into a growth phase. And then at the second telogen, it actually sits pretty quiescent for about 6 to 8 weeks. And this is very exciting for those of us working in the stem cell field, because this is a really good opportunity for us to isolate these cells at this stage. And then when we went back and looked at the knockout, what we could see was that the morphogenesis was reduced. There was a longer catagen. It actually had a sort of telogen. And then it immediately went back into morphogenesis, catagen, and telogen, so that by day 35 to 37, it was already sitting in its telogen, and then it became asynchronized. So this hair follicle cycle was extraordinarily fast compared to what we saw in the wild type. So what I've told you so far is that the conditional knockout of Vinculin results in a very sparse hair and accelerated hair follicle cycle. There is a very short transient telogen phase or quiescent phase, and of course the hair follicles are smaller. So Vinculin, of course, is a protein that's associated with junctions. So how might the loss of Vinculin, in fact, explain some of these effects? So as I mentioned before, Vinculin, of course, is present in both these junctions, where in both cases functions as a mechanotransducer. So is it the cell substratum function of Vinculin, or is it the cell-to-cell function of Vinculin that was important for this process? And so we decided to address one at a time. So we initially looked at the cell-to-ECM junctions. And what we saw was that there was actually no overt effects. So we looked at the expression of extracellular matrix proteins. In this case, we looked at collagen 4, and compared to the control, the knockout actually had pretty reasonable looking ECM. Expression of collagen 4 at its extracellular matrix boundary. And when we looked by EMs, what we could see very nicely was that there was a very nice ECM. This is sort of the acellular layer that I was mentioning before. And you can see that, and you can also see the junctions known as the hemidesmosomes that are being formed quite nicely in the Vinculin knockout. So that for us ruled out sort of an overt cell to substratum junction phenotype in this hair cycling defects that we were seeing. So we next focused on the cell-to-cell junctions, which are the adherence junctions. Now, adherence junctions, as I mentioned before, are cell-to-cell junctions that are nucleated by this homophilic cell adhesion molecule called E-cadherin. So E-cadherin on its extracellular side binds to another E-cadherin molecule, and this is calcium dependent. So in the presence of calcium, there will be cell-to-cell junctions formed. And then on the cytoplasmic side, E-cadherin will recruit a bunch of proteins known as catenins, so P120 and beta-catenin. Beta-catenin is pretty famous because this functions also as a transcription factor downstream of Wnt signaling. And then beta-catenin recruits this protein called alpha-catenin, and alpha-catenin will recruit this protein called Vinculin. So in the normal course, both alpha-catenin and Vinculin live in sort of this folded state. And as I told you before, once they experience forces, they unwind. And as they unwind, they're able to associate with the actin cytoskeleton network, which is represented here. And so so this is, again, just the schematic of what I was telling you, which is you have sort of the activated Vinculin and the stretched alpha-catenin, which associates with the actin cytoskeleton network. So what happens when we remove Vinculin from these cell-to-cell junctions? So again, let me just walk you through these slides. So this is this is the control, and this is the knockout panel. So this is the first telogen and then the second telogen. So we looked at both telogen phases because this is the place, remember, where the junctions are the most stable. And so what we could tell right away was in the wild type, these junctions were formed quite robustly. And in the knockout, we found these junctions to be quite messed up. And that was true whether we looked at E-cadherin or other proteins such as alpha-catenin, which I'm not showing you here. So what was also very interesting is when we went back and looked at these junctions in the bulb stem cell compartment, what we were able to show both in the first and the second telogen was that instead of forming these tight cell-to-cell appositions, which is marked by desmosomes, in the Vinculin knockout, we could see sort of these separations in the plasma membrane between cells. And this was found both at the first and second telogen, and this was associated with quite a significant decrease in the total number of desmosomes. And remember, I told you desmosomes we're using as a proxy to study the number of adherence junctions. So this data suggested to us that in fact, in this bulk stem cell compartment, in the so-called quiescent bulk stem cell compartment, in fact, the number of cell-to-cell junctions was dramatically reduced. And so what this told us was that in fact, Vinculin seems to play a very important role in maintaining these cell-to-cell junctions. So to test this further, what we decided to do was we decided to use a calcium switch assay. And this is an assay where, remember I told you cadherin junctions are dependent on extracellular calcium. And so when you provide cells with extracellular calcium, you form these stable cell-to-cell junctions. And so this is just the wild type. You can see very beautiful formation of cell-to-cell junctions. And in the knockout, you could see that the junctions were not zippering up. And that was the same when we looked for this other protein, alphacatenin. You could see right away that the junctions were not zippering up. And so then when we did scanning electron micrographs of these cells, what we could see was that, in fact, the wild type was beautifully zippering up. They send out these actin structures, these philopodial structures that help to zipper up the two junctions. And the knockout was just, these junctions were not formed normally at all. And so we then decided to just look at this using live cell imaging. So these are just wild type and knockout keratinocytes that have been transfected with an E-cadherin GFP. And so on the top panel, you can see that the wild type cells, when they come together, they form this beautiful cell-to-cell junction. So what you would notice is cadherins remain cytoplasmic, and the minute you form a cell-to-cell junction, these become stable at the cell-to-cell junction. Whereas with the knockout, what you can see is that firstly, the cells are much smaller. They have a spreading defect. The other thing is that they're highly migratory, and then the junctions are very labile. So they seem to be able to form junctions and they break it up right away. So again, this suggested that these junctions that these Vinculin knockout cells are forming are quite weak. So we then wanted to ask, okay, what happens to alphacatenin? Because of course, in these Vinculin knockout cells, there should be alphacatenin expressed. I've shown you that there's alphacatenin expressed. So how come, what is happening? What is the role of alphacatenin in all of this process? And remember, alphacatenin also associates with Vinculin, and Vinculin is very important for the stabilization of alphacatenin. And so, in order to look at that, we collaborated with my friend and colleague, Valera Vasiukin's lab. And he generated an alphacatenin conditional knockout. And what he did was he only removed alphacatenin from the bulge stem cell compartment, shown here. So this is in green as the alphacatenin expression, red is the cadherin. You can see that alphacatenin is only missing from the bulge compartment. It's present in the rest. And so already you can see that these animals are completely bald, suggesting again that that the loss of alphacatenin has a critical defect in hair morphogenesis. And when we went back and looked at the hair follicle cycling, particularly in the second cycle, what we could see was that there was a dramatic difference in the way these hair follicles were growing out. In fact, these follicles failed to grow out completely. And that would probably explain why these animals were eventually losing their hair. We also looked at the junction formation, and we were able to show that, in fact, the junctions were completely messed up. These are the cadherin-based junctions in the alphacatenin knockout. Alphacatenin knockout keratinocytes, of course, don't express alpha-cat. And very interestingly, Vinculin fails to localize to the cell-to-cell junctions. It's only found in cell substratum junctions. So again, what was interesting is that this other actin-associated protein that's present in the adherence junction seemed to have very similar phenotypes or slightly worse phenotypes than Vinculin, vis-a-vis the hair follicle cycling and the cell-to-cell junctions that were being formed in these hair follicles. And so the question that we asked then was, why are these intracellular junctions that are being formed so weak? Right? Because clearly they're being formed, but they're not being able to be maintained. And so what is the forces that are being generated by these cell-to-cell junctions? So in order to address this, we collaborated with Yan Ji's lab at the Mechanobiology Institute here in Singapore. And he came up with a really ingenious way of measuring this. And so what he did was he had this microscopy set up. And essentially what you do is you take these beads. So this little black dot here is a bead. It's a 3-micron bead, which is now coated with ecadherin. And what you do is you put it onto the cells and you let it attach for about 5 minutes. And then you take this glass micropipette and capture the bead, and then you move the stage on which these cells are sitting in a dish in a stepwise manner. And then when the beads actually detach from this glass pipette is sort of a function of the adhesion strength of these beads to the underlying cell, right? Because it's the cadherin based. And so one can calculate the forces that are generated by these junctions using just a formula over here, which is basically just the traveling distance as well as the bending constant of the micropipette. And so this was done using both the wild type and the knockout cells. And so we wanted to ask, okay, so what happens when you lose Vinculin? What are the forces that are being generated by these knockout cells? And very interestingly, what you could show was that the bead detachment, so remember we're measuring the forces that are required to detach these beads. And so for the wild type, this was about, the amount of force that was required was about 6 nanonewtons. And for the knockout, the bead sort of detached in less than half of that force, about 2.5 nanonewtons is all it took to detach the force. And interestingly, this was completely dependent on Vinculin, because when we added back Vinculin to the knockout cells using a GFP construct, we were able to completely rescue the force generation by these cells. Again, suggesting the absence of Vinculin was critical, had critical failure in maintaining strong cell-to-cell junctions. And so when we go back and look at what these junctions look like, this is just the control. And in the knockout, what you can see, as I told you, there is quite a bit of alpha-catenin expressed, but this is completely messed up. And of course, there is no Vinculin that's expressed in the Vinculin, in the knockout. And then when you have the rescue, you come back very nicely. You're able to rescue both the junctional defects as well as the cell substratum defects. What was really fascinating to us was that, in fact, in the Vinculin knockout, there was not only the expression of cadherins, but also an overexpression. So there was a overexpression of ecadherin and alpha-catenin, and some overexpression of beta-catenin, although it was not significant. So what this told us is that, in fact, the cells are kind of sensing that there is an issue with the cell-to-cell junction, and they're trying to compensate for this by generating more of these cell adhesion molecules, but it doesn't seem to be making much of a difference. So then we wanted to ask what happens to the alpha-catenin knockout cells. Remember, those are also cells that don't make normal cell-to-cell junctions. And so very similar to what we saw with the Vinculin knockout, the wild type, again, has a pretty high detachment force. The alpha-catenin knockout has about half of that. And when we add back alpha-catenin, and these are just the cells showing all of this, you can actually rescue back the junctional forces. Now, what we decided to do was we decided to rescue alpha-catenin with this particular construct, and this is called the L344P catenin construct. And what this is basically, it's an alpha-catenin that fails to bind Vinculin. Okay, so the hypothesis here is that if Vinculin is important for the unbinding of alpha-catenin, and it's important for the force generation, then if you put back, even if you put back alpha-catenin without its ability to recruit Vinculin, it'll be like having a Vinculin knockout. And that's exactly what we see. So this L344P mutant is unable to generate forces, and even though it's recruited to the cell-to-cell junctions. Again, suggesting that Vinculin plays a critical role in functioning as a force multiplier as far as force generation is concerned at these junctions. And so what I've told you so far is that the wild type have a normal cell-to-cell junction, and they have a relative force of about 6 nanonewtons, and the knockout have abnormal junctions and have a relative force of about 2.5 nanonewtons. So, okay, so we wanted to ask next, what is the mechanism by which this force generation is happening, and what are the consequences of this sort of loss of junctional stability. And so to do that, we decided to isolate the bulk stem cells from the knockout and the wild type animals and subject them to next generation sequencing. And one of the major pathways that was upregulated in these knockouts was the targets of the hippo-YAP pathway. So I'm sure all of you have heard of the hippo-YAP pathway. This is a growth control pathway, which is controlled by a kinase called hippo. And one of the targets of this is this protein called YAP, which then translocates to the nucleus. And one of the things it does when it translocates to the nucleus is it turns on cell cycle regulated genes or cell cycle genes. So a lot of the cyclins are downstream of YAP signaling. And what's interesting with TAS, YAP and its cousin TAS, is that not only does it work downstream of in a hippo-dependent manner, but it also works in a hippo-independent manner, and that is just by force sensing. So any forces that are sensed from at the level of the cell substratum or cell-to-cell junction impinge on the localization of YAP into the nucleus and also the proliferation state. So if a cell is sitting in a very soft matrix, YAP does not translocate into the nucleus, and if it's sitting in a stiffer matrix, YAP then translocates into the nucleus. And so we wanted to go back and ask what is happening to YAP in both the telogen phase of the hair cycle, as well as in confluent cells in culture. So again, let me just walk you through this. This is the expression of YAP in telogen, in controlled telogen follicles. And what you can see is YAP is actually present at the cell-to-cell junctions. And interestingly, in the knockout, in the so-called quiescent phase, YAP is strongly sitting inside the nucleus. And this is just quantified over here. And likewise, when you grow cells at very, very high confluence so that they're contact inhibited, very much like the bulk stem cell compartment, YAP is sitting in the cytoplasm of these cells. And in the knockout, what you can see is that YAP, even though the culture is highly confluent, these cells have YAP sitting in the nucleus. So one of the phenotypes of the Vinculin knockout is that even though we grow them to confluence, they keep continuing to grow. So they're really non-contact inhibited. So they're no longer able to sense their nearest neighbor, and they keep proliferating because YAP is sitting inside the nucleus. And so what is the consequence, of course, of having all this YAP in the nucleus? So one of the consequences is just the loss of stem cell quiescence, right? And so we can look at this by the expression of the stem cell marker CD34. And what you can see is in the first telogen, you still see some expression of CD34. In fact, that's why we were able to even isolate these stem cells. But by the time these hair follicles reach the second telogen, there's a complete loss of CD34 expression. And this is accompanied by the loss of the long-term label retaining cells. So if you mark cells with BRDU or EDU, you can chase them for a long period of time. These are still EDU positive cells in the control. And these EDU positive cells are completely lost within the knockout. And concomitant to the loss of EDU is the increase in proliferation potential. So you can see increase in Ki67 levels in the so-called telogen follicles compared to the controls. So again, what this told us was that the loss of the junctional stability in this was pushing YAP into the nucleus, and that YAP was now driving the cell cycle and increasing the proliferative potential of these stem cells. And so, of course, so what is the mechanism by which this is happening? So it turns out that, in fact, alphacatenin, which is your other junctional protein, can actually sequester YAP at the junctions by its association with this protein called 1433, which is an adaptive protein. So under conditions of very high density, alphacatenin will keep YAP at the junctions because in a 1433 dependent manner. But where the cells are either in very low density, such that they're no longer forming cell-to-cell junctions, or when you have removed alphacatenin such that you don't form cell-to-cell junctions, again, YAP 1433 is no longer able to sequester, and then you basically get a dephosphorylation event, and YAP, the unphosphorylated YAP, actually goes into the nucleus to basically affect target gene expression, such as the cyclins. And so So we know that alpha-catenin sequesters YAP in the cytoplasm. But of course, the question was that why isn't alpha-catenin able to hold on to YAP? Because alpha-catenin is there, right? Remember, I told you alpha-catenin is there in the Vinculin knockout. There's in fact high levels of expression of alpha-catenin, but why isn't it able to hold on to YAP? So it turns out that alpha-catenin also, very much like Benkillin, is force activated. So it lives in this closed conformation, and then it requires approximately 5 piconewtons of force and the presence of Benkillin to be able to open up into its activated state. And when it's in this active conformation, it is actually recognized by a specific antibody known as the alpha-18 antibody. And so we looked at this conformation-specific antibody staining within the knockout and the wild type. So remember, alpha-18 is what marks the open conformation. So, and alpha-catenin antibody just marks the total levels of alpha-cat. So in the control, there is equal expression of alpha-catenin, alpha-18 as shown here and basically quantified here. And in the knockout, even though there's very high levels of alpha-catenin, the alpha-18, which is the open conformation antibody, is no longer expressed. And that is also shown here. I'm not able to see the side of my graph, but I'm sure you can see that. So it's quantified where the levels of alpha-18, even though tons of alpha-catenin is present, there's no alpha-18. So likewise, when we looked at the cell-to-cell junctions for the expression of alpha-18, we found that it was actually very poorly expressed. And then we actually did a 3D SIM imaging, and where we were able to show that, in fact, the alpha-18, which normally associates with the actin cytoskeleton network because it's in this open conformation, was very beautifully associated with the actin in the control, but in the knockout was no longer expressed or no longer associated with the actin cytoskeleton network. So this suggested that in the absence of Vinculin, alpha-catenin was no longer in this open conformation. And so because we know alpha-catenin sequesters YAP, and I told you that this is through 1433, we went back and asked what's happening to YAP. And so in the presence or in conjunction with the alpha-18, which is this open conformation antibody. So when we go back and look at alpha-18, and at the presence of YAP, so YAP is at the junctions along with alpha-18. In the knockout, YAP is all in the nucleus, and there's very little expression of the alpha-18 or the open conformation antibody. Likewise, in keratinocytes, what we could show was that if we re-express Vinculin in the keratinocytes and the knockout keratinocytes, we are able to bring back alpha-18 to the junctions. And this is enough to kind of exclude YAP from the nucleus at this point. Again, suggesting there's a very critical correlation between, or a critical interaction between YAP and alpha-catenin. And because alpha-catenin is not in this open conformation in the Vinculin knockout, even though tons of it is there, in fact, threefold expression of alpha-cat is there, but because it's not in the correct conformation, it's not able to hold on to YAP. And because it's not able to hold on to YAP, YAP is free to go into the nucleus and mediate cell proliferation. And so, of course, YAP is held at the junctions through 1433. So we looked at the expression of 1433 and YAP, and you can see that they're actually co-localized in the control, but in the knockout, the 1433 has a more diffuse pattern of expression. So this brings us to the model of what I just talked to you about, which is that in the control, the cell-to-cell junctions are nucleated by the cell adhesion protein, homophilic cell adhesion protein called E-cadherin. And E-cadherin will recruit, of course, the series of catenins. And then you have, when you have Vinculin present, Vinculin, one of the critical roles of Vinculin is not just associating with the actin cytoskeleton network, functioning as a force multiplier, but also to keep this protein alpha-catenin in this stretched conformation. And when it's in the stretched conformation, it's able to hold on to YAP at the junctions through its association with this adaptive protein 1433. So in the conditional knockout, what happens is that there is no Vinculin. So now alpha-catenin now folds back on itself. And of course, remember, I showed you this by the alpha-18 conformation specific antibody staining. And when this happens, 1433 then is no longer able to associate because the binding site for 1433 alpha-catenin is now blocked. And so because it's no longer able to associate, YAP is now free to be free to be pushed into the nucleus. And then when it's pushed into the nucleus, it actually affects proliferation by controlling cell cycle regulated genes. The loss of Vinculin also, and in fact also results in the loss of alpha-catenin. And because you're not generating the forces that are required to keep these junctions together, the junctions also start failing. And so you also have this sort of loose junction that's associated with sort of the intracellular forces that have to be generated by Vinculin and also by alpha-catenin because Vinculin is no longer there. And so to summarize what I told you today is that the wild type pulse stem cells have normal cell junctions. These are contact inhibited. You can see this in the phalloidin staining that you can see within the pulse stem cell compartment. They have these beautiful hexagonal shaped cells, which are contact inhibited, quiescent, and have cytoplasmic gap. And in the knockout, these have abnormal cell junctions. These are non-contact inhibited, non-quiescent, and they increase the level of nuclear lab, basically providing a mechanism for loss of quiescence. And so the reduced intracellular adhesion, in fact, can override what we call the intrinsic quiescence factors. So suggesting that, that junctional strength is critical. And this is important not just, of course, for the bulk stem cell compartment, but as you know, that in many of the cancers, they actually regulate or downregulate cell junctional strength. And in fact, cancers, aggressive cancers are associated with very high YAP activity and very low E-cadherin activity. So we think that these kind of mechanisms that we've described using the skin as a model system really can be translated to other systems where, there's a YAP-dependent cell proliferation that's happening. And so if you want more information about the story that I just told you, this was just published last year. And so in the last 5 minutes of my talk, or last couple of minutes of my talk, what I thought I'll tell you about is where we're going from here, right? So we've already described this idea that the junctions are critical in maintaining cell fates and cell states, in this case, the stem cell compartment. And so we wanted to ask, okay, in some sense, where does one go from here in terms of, extending the story? So we got very interested in understanding sort of the link between junctions, both cell-to-cell junctions and substratum junctions, and the nucleus, specifically looking at, the nuclear membrane and complexes of protein that are present at the nuclear periphery. And the reason for this is that it is becoming increasingly clear that, mechanical forces play a really important role, not just in development, but also in disease. So one example for this is, diseases like cardiomyopathy, right? Because when the cells are no longer able to sense the forces correctly, the cells become hyperproliferative and they change their ECM, the type and quality of the ECM that they produce, leading to fibrosis and diseases. And so we got very interested in this. And partly the question that we wanted to ask was that, what is the crosstalk that occurs between junctions, at the cell periphery and then nuclear proteins? And so we're actually using a genetic approach to do this. So of course, at the cell periphery, I just explained to you this whole story where we see that, Vinculin is of course present at both these junctions, and it's of course a mechanotransducer. And at the level of the nuclear membrane, we're looking at these link complex proteins, specifically both SUN1, SUN2, and the double knockout. Although today I'll only talk to you a little bit about SUN2. And so why did we get interested in this? So my postdoc, Avi, who came from a nuclear mechanics lab, basically had this following observation that in the Vinculin knockout keratinocytes, she found that about 60% of the nuclei appear crumpled. And this is just sort of, again, a 3D sim image showing that these nuclei are really crumpled. And I have to hasten to tell you that these are actually not pathological nuclei. These are just nuclei that look like raisins. They're just crumpled, right? And so, okay, what does this actually mean? So, of course, as I told you, as I've been telling you throughout the course of this talk, that Benkillin is present at both junctions, it's present at the adherence junctions, it's present at the focal adhesions. So is this crumpling or raisin-like phenotype of the nucleus, what does it, does it have to do with defects at the level of the adherence junctions or defects at the level of the focal adhesions. So we decided to look at this by bringing in a couple of other knockouts. Alphacatenin I've already talked to you about. Alphacatenin is exclusively present only at the adherence junction. And then the other knockout we brought in was the beta-1 integrin knockout, and beta-1 integrin is present exclusively at the focal adhesions. And the idea was, by comparing these various the nuclear phenotypes and the nuclear morphology of these various knockout cells, we could ask if this wrinkling or crumpling that we see is a function of Vinculin at adherence junctions or focal adhesions. And so to do that, we started looking at nuclear lamins. Shravanti will tell, I'm sure you've heard a lot from Shravanti about nuclear lamins, but basically they're just, these are intermediate filaments that are found at the tip of the nucleoplasm. There are two types of lamins. There's lamin A and there's lamin AC, and then there's lamin B. In the absence of lamin A, you have lamin B that can take over. And of course, the loss of lamin A will leads to a disease called progeria. And so, or the premature aging disease. And so we went back and looked at the expression of lamin AC. And what we could see, so these are just, a confluent monolayer of cells. So in the wild type, we saw this beautiful peripheral expression of lamin AC. And interestingly, in the beta-1 integrin knockout, also we saw a very beautiful peripheral expression of lamin AC, whereas in both in the Benkillin and the alpha-catenin knockout, we saw this sort of crumpled effect, if you will, which is all quantified here. Likewise, when we looked at lamin B, we saw the same thing. So the wild type and the knockout, so the wild type and the beta-1 knockout had beautiful peripheral expression, and the knockout had sort of this crumpling happening. So, I've already told you previously that in fact, the perturbations, the Vinculin has perturbed cell-to-cell junctions, right? And so what I'm telling you now is that the loss of cell-to-cell junctions, whether it is through Vinculin or alpha-catenin, is what seems to be responsible for this nuclear crumpling. Because if you lose cell-substratum junctions, or if you perturb cell-substratum junctions, that doesn't seem to be the case. So then what happens on the other end? What happens if you perturb the link complex? So in this case, we knocked out SUN2. And so again, the link complex are known as a linker of nuclear skeleton and cytoskeleton. So as the name implies, it forms a connection between nuclear lamins and the cellular cytoskeleton through this other set of proteins known as mesprins. There are two SUN domain proteins present, SUN1 and SUN2, and they're really important, have very critical roles in nuclear envelopes as well as cytoplasmic forces. So in the SUN2 knockout, very fascinatingly and very interestingly, what we found was that in fact, both lamin A, C, as well as lamin B expression was, indicated that the nuclei in these cells were also crumpled. And also quite fascinatingly, the cell-to-cell junctions were also quite perturbed. So of course, you could just say, Srikala, this is all just correlation at this point, right? I mean, you're finding two sort of, the two ends of the spectrum having very similar phenotypes. And I think I completely agree at this point, it's just very tantalizing correlations. But except to say that we've actually done NGS analysis of the Vinculin knockout, and one of the things that does get affected are some of the link complex proteins, right? So our idea then is, we're very interested in understanding how does the cell junction talk to the nucleus? How does the nucleus sense the cell junctions and sense the forces through the cell junctions? And how is this crosstalk actually happening through mechanobiology, through cell mechanics? And ultimately, of course, all of this impinges on transcriptional readouts. And so what does this crumpled nucleus actually mean? Does it mean that these, of course, we know that these cells are more transcriptionally active. So what does it mean vis-a-vis its chromatin landscape and its organization? So this is a project that we've embarked on in collaboration with Brian Burke's lab. And as I said, we're going to be using a genetic approach to actually look at all of this. And so this is where we're going with this project. And so now it all just remains for me to thank everybody that did this work. And so all of the work I talked to you about today, the Benkillin work, was done by an outstanding graduate student, Ritushri. Actually, she's just defended, so she's Dr. Ritushri Bishwas. And my postdoc, my amazing postdoc, Avinanda Banerjee. So the two of them were this amazing team that worked on this. Sergio was a master's student in the lab. He came from IFA Milan and spent a year and just was a force of nature and did amazing work. And my lab in Singapore, this work is now being, so Avinanda moved here, and we have Joy, who has joined our group, and she's continuing to work on some aspects of the Sun1 and Sun2 knockouts. And Ryan is also initially worked on some of this. Vairavan helped us with the NGS analysis, and Sihai helped us with the the force measurements. I have a bunch of really incredible collaborators who've collaborated with us on this project. Also want to thank our funding sources and facilities, both at InSTEM as well as A-STAR. And with that, I thank you for your attention, and I'm super happy to take questions.
Thank you, Srikala. That was a wonderful talk. So there is one question in the chat box. If you can, Pooja, if you can read it out, and people can raise their hands as well if they have any further questions.
So we have a question from Kanan. He says, what happens to the overexpression of Vinculin alpha-catenin? And also, what is the link between hair formation or development versus Vinculin and alpha-catenin? The following question is it possible to culture these stem cells in vitro? If yes, do they require hair?
Okay, so let me take it one at a time. So what happens when you overexpress? Is that the question?
Yes.
You overexpress Vinculin and alpha-catenin. So, so actually, very interestingly, at least with respect to the skin, when we try to overexpress these proteins, we don't see, so for example, you remember we introduced back Benkillin, right, into the keratinocytes, and we're not actually expressing it at physiological levels, not always. We hope we are, but we're not always expressing it at physiological levels, but it seems to be bringing back the phenotype, and we don't see any adverse effect with higher levels of expression. My sense is there's some level of regulation of the actual protein expression, which is done by the cell itself, so it's only going to use as much as it needs. Yes, we can culture these cells. And so all of the keratinocytes that I showed you were actually stem cells that were coming out of either the Vinculin or the alpha-catenin knockout. And yes, they behave the same way. They're non-contact inhibited. They're highly proliferative. What I didn't show you with what's in the paper is that these guys, in fact, don't really function as stem cells, because if you take them out of their niche and ask them to form hair and skin, remember I told you that the bulk stem cells are multipotent. Under conditions of wounding, they can form not only hair, but skin. And so when we try to do these reconstitution assays, while the wild type is able to make beautiful hair and skin, the knockout is unable to do so. And so, even though we call them stem cells, I think they no longer have the capacity to function as multipotent stem cells. And what was the other question?
He also asked, what is the link between hair formation or development versus Vinculin and alpha-catenin?
Right. So I think what our data is suggesting is that it's not so much the hair formation, because the hair formation is quite fine, as you can see. The hairs are forming. There's less hair, but it is forming. But where Vinculin and alpha-catenin are critical is in maintaining the niche of the stem cells, right? So what our work is suggesting is that it's not just cell intrinsic factors that are important for this, for maintaining the niche, but also these junctional proteins are critical. And, the niche, the stem cell compartment really looks to be very contact inhibited. And so if you don't have these proteins at that stage, then you're not able to maintain a normal stem cell compartment. And that has profound effects on maintaining stemness. And so that was kind of exciting for us, because, all these 30 years, people have been telling us that in fact, it's the intrinsic factors that are really critical, right? People have been knocking out these cell intrinsic factors and shown that they're very critical for stem cell maintenance. But while what we're saying is not only those, but also cell junctions are critically important for this process.
Thank you for addressing that, Dr. Raghavan. Along the same lines, I myself have one question. So in case of alopecia and patients who are losing hair due to chemotherapy, so currently we have FUE and FUT technologies for hair transplantation, but What are the challenges do we face in case of going for a bulk stem cell transplant in that case of alopecia especially? So what do you think are the challenges and what should be the strategic line in case if you're talking about this, the cells being losing of the stemness? So if we could use modulators for vinculin or alphacatenin during that transplantation strategies. So what are your
Yeah,
views
that's
on that?
very interesting. So, so the two completely separate issues. So one is hair transplant, presumably in just regular folks, right, where you're losing, female pattern baldness, for example. The other is alopecia. Alopecia is an immune disorder, right? It is basically an autoimmune disorder where the immune system attacks the hair follicles and essentially causes a destruction of the hair follicles. So the way to control alopecia has really been to control the immune response. And that has actually been quite effective. In fact, there's some very effective therapies, immunomodulators that are being used to control alopecia. And so when people are on those, then they're actually able to get back hair follicles. Their hair grows back. So it's just the microenvironment around the hair follicle is very hostile for the hair follicle, and then basically it destroys it. In terms of transplantation, so it's very interesting. So hair transplantation is a huge industry. It's a multi-billion-dollar industry, right? People want, and so what's interesting is that if you take obviously, you have to transplant the whole hair with the bulge compartment. And what has been shown is that particularly, with male pattern baldness, when you take hair and you transplant it into a new spot, the hair actually will grow back. But interestingly, it maintains the memory of where it came from. Not the new, it doesn't acquire the fate of where it is. And so to give you an example, so people usually have this male pattern baldness where they lose their hair at the top of their head. And so very often, so what happens is people will take, sort of follicles from the back of the hair and transplant it to the front. And so there is a difference in sort of, thickness of hair follicles between even different sites within the scalp itself. And so when you do that, then those follicles look more like from the back of the head rather than from the top of the head. And so, and the other interesting thing with hair transplant, in fact, is, and our colleague Shravanti and my colleague Colin has this beautiful study out last year where what they had done was, this was in collaboration with some dermatologists, where they had taken hair follicles from patients who had non-healing diabetic foot ulcers, right? So diabetes is a huge issue in India, and many patients will end up getting these non-healing wounds, and there's very little you can do for them because there's just the wound healing capacity of the skin is highly compromised in diabetes. And so what they showed was that they can put these hair follicles, they can implant these hair follicles at a specific at a specific distance, and these hair follicles are able to sort of, the cells are able to contribute not just to the skin, so they reepithelialize the skin, but they're also able to, reform the blood supply, because that's one of the issues with diabetes is that the blood supply goes away, and also some of the nerves. So it's a really beautiful story that just came out in JID. So it's two different issues, but I think that is why it's really exciting. So there's a huge potential for using these hair follicles and the stem cells that lie within it for further therapeutics, not just in recreating hair. We don't need hair. I mean, we really don't. It's not important for our survival. But skin is important for our survival. If you don't have skin, then you're not going to make it, right? And so there's a huge amount of interest in using these or leveraging these stem cells for those kind of therapeutics.
Thank you, Dr. Raghavan. So we have one more question from Dhruv. He asks, what happens to nuclear architecture, cell junctions, when you perturb actin and tubulin cytoskeleton?
Yeah, that's a really good question. So yes, so in fact, when you perturb the actin cytoskeleton network, you actually do see changes in the nuclear architecture or the changes. So you see these cellular types of crumpling. And with the tubulin, I'm not so sure. We haven't actually done that yet. But I would presume that it's also going to have an effect. So what this suggests is that there are forces that are being sensed through the actomyosin contractility network to the nuclear periphery from the cell periphery, and that force is being transmitted. It's not something that I have discovered or that I'm telling you that's something new. This is something that if you attend mechanobiology talks, they will tell you ad nauseum, this is what is happening. The question is, what does it actually mean, right? And one can say, oh, there are all these forces, but does it have any biological relevance? And the answer to that is yes. We really think it has some very critical biological functions.
We have one more question, Dr. Raghavan. It's from Sundaram, so I'll just read it out. You showed that junction got weakened in both vinculin and alpha-catenin knockout cells than control. However, there was still quite a good amount of residual junctional strength retained. I'm wondering, what are the other players to maintain this much of cellular connection? Also, have you seen what happened where both vinculin and alpha-catenin are knocked out?
Yes, really good question. So, and that's a really good question. So I think, remember that, that even though the absence of absence of Benkillin can really perturb cell-to-cell junctions, the desmosomes are still being formed, right? So it's not that there is no junctions being formed. So the residual junctional strength that we're seeing, whatever we're seeing is from the fact that, in fact, for E-cadherin to be localized to the junctions also requires the formation of these desmosomes, and the desmosomes also require desmosomal formation also require the formation of adherence junctions. So they both kind of are mutually beneficial for each other. And so cadherins, even though they are not formed correctly, they probably provide enough of a junctional strength in order to provide about 2.5. So the only way you will not get any cadherins at all at the cell surface is if there's no calcium. And we do that routinely with skin cells, right? So if you put them in low calcium, there are no junctions formed and there's no E-cadherin on the cell surface at all. And so that is, but then that's not the case here. There is E-cadherin. It may not be, it may be more labile, it may be turning over faster, but it is still there at the cell surface. So it is forming some sort of junctional strength. So it's not that it's completely fallen apart. Alphacatenin and Benkillin. So the alphacatenin conditional knockout is neonatal lethal. Because those cells just fall apart. So that is exactly what. So in fact, that would explain to some extent what happens in a double knockout, because in the alpha-cat knockout, there's no Vinculin at the junctions. So in those cases, if you look at the skin, it's just individual cells that you see. There is no stable cell-to-cell junction being formed.
Oh, that is informative. I hope. So with the interest
I have a
of
question. If nobody has a question, can I?
yeah, sure.
Every time I listen to her talk, I always have a question for her. Srikala, I was wondering about this Benkillin and catenin generated forces. So what happens in the context of aging? Because you seem to suggest that the forces actually comes from the substratum, right? And because of during aging, because this collagens and everything fall apart, do you think that is responsible for loss of bulk stem cells and loss of hair appendages?
So in fact, levels of Benkillin reduce upon aging. So
okay.
Yeah. So there is that, but the loss of So if you have male pattern baldness, it is not because of the loss of Vinculin. It's actually, again, it's because there is sort of an antagonistic effect from androgens that prevent the formation.
Yeah,
and
that is hormonal.
So that is hormonal.
Yeah.
And so there are people who are very old that have a full head of hair, and are perfectly fine, right? But the levels of Vinculin do reduce upon aging. And what you have beautifully shown is that the nuclear morphology also gets squishy on aging. And I think there is something to that. So I really think upon aging, the junctional, even minor perturbances in the junctions are enough to change sort of nuclear morphologies, or nuclear morphology may affect junctions. It could be both ways. It doesn't have to be a single way, right? We think it's both are talking to each other.
It could be inside out.
It's inside out and outside in, yeah. So it's happening both ways. And I think, so all of these pieces of data that are all disparate are just very tantalizing and would suggest that in fact, because you have to think that a cell that is sitting, a nucleus that is sitting will have to get information, not just, what it has to transcribe, but also, it has to get some signals on what its local environment is. I'm thinking like a nucleus, but it needs to start sensing. It has to sense where it is. And we also know from the cancer literature that not having these junctions has profound consequences on nucleus shape. So in fact, very metastatic cells, and in fact, we have actually done those experiments where we've taken very metastatic cells which don't have cell junctions, and the nucleus are completely squishy. And we know that, it's again been described beautifully in the literature, but what does that actually mean? What does it mean to have a squishy nucleus, right? Other than the fact that it can actually migrate beautifully through capillaries, and they've shown all these gorgeous movies, all the mechanobio people, put it into tubes and show you all of this, but what does it actually mean? What does it mean to have this squishy nucleus? And to me, and my very simplistic way of thinking about it is, it's just, you are making available parts of the chromatin is just more, is more available for transcriptional activity. And you're just making the nucleus much more permissive for transcriptional activity compared to, say, a more differentiated nucleus where all the heterochromatin is sitting on the cell periphery. And so we're going to embark on high-C experiments to start looking at some of that.
Okay. It was really an exciting talk, Srikala. How many ever times I hear that, I always love the work.
Thank you.
So if there are no more questions, I guess, Pooja, do you want to conclude the talk?
Yeah, I think we have to keep in interest with the time as well. So thank you so much, Dr. Raghavan, for addressing all the queries and questions. It was a fantastic talk and was highly informative for all of us who've joined. Thank you for coming and taking the time out. And over to you, Dr. Rampalli.
Yeah, thanks, Srikala. This was really great. And whenever you'd come to India and specifically Delhi, we would really like to invite you to IGIB to talk in person.
Yeah, I'll be there next time. Absolutely. I anyway have to come and see you, so I will come.
Yes, thank you. Thank you, and have a good night. And
you
thank
too.
you.
Have a good evening, all of you. Thank you so much.
Bye. Bye.
Bye.
Bye.
Bye.